Most of us first met the cell as a flat, brightly colored diagram in a high school biology textbook. We were taught to view it as a loose bag of fluid containing a random assortment of floating shapes, and our job was simply to memorize the names of those shapes. But that static model is deeply misleading. Trying to understand how a cell actually functions by looking at a disconnected list of parts is like trying to understand how a car drives by staring at a pile of spark plugs and belts laid out on a garage floor. The reality is highly active, starting at the very edge. The cell membrane acts as a flexible phospholipid boundary, functioning as a pressure-sealed container for a massive, continuous chemical chain reaction. A membrane rupture triggers cell death, because it destroys the physical environment required for internal logistics. When the fluid disperses, the cell can no longer maintain the pressure or chemical concentration needed to sustain its coordinated sequences. To actually understand a biological machine inside us, we have to throw out the textbook diagram and watch the system in motion. We have to trace the continuous, logical life cycle of a single protein. Inside the nucleus, DNA unzips, creating messenger RNA. This mRNA feeds through a ribosome, linking amino acids into a chain. The rough endoplasmic reticulum folds this chain into a 3D shape. Then the Golgi apparatus packages it securely inside a vesicle, ready for transport. This vesicle acts as a shipping container. It carries the finished protein where it's needed, fusing with the cell membrane to dump the protein outside the cell, a process called exocytosis, or delivering it to another internal structure like the smooth ER to help synthesize lipids. Building a protein is a relay race. It is a continuous, interdependent sequence passing seamlessly through 5 distinct organelles. But running a factory of this scale poses a logistical problem. Physically linking amino acids, folding complex shapes, and shipping vesicles requires an immense amount of work. Where does the cell get the power to do all of this? It relies on the mitochondria. Through a process called cellular respiration, the mitochondria takes in high-energy fuel from our environment, specifically the sugar we eat and the oxygen we breathe, and systematically breaks those chemical bonds apart. In this visualization, we see exactly how that energy is captured. By snapping those larger molecules apart and reforming them into lower-energy byproducts like water and carbon dioxide, the mitochondria captures the released energy. It repackages that energy into a universal currency called ATP. When people call the mitochondria the powerhouse of the cell, this is the mechanical reality behind the phrase. The ATP it generates is the direct physical fuel used by the internal pumps and transport channels to push our protein through its manufacturing journey. Operating a high-speed manufacturing pipeline and burning constant energy inevitably creates hazardous chemical waste, and the rich resources inside the cell attract external threats like pathogens. To manage this, the cell relies on lysosomes. These organelles operate like microscopic recycling centers and defense mechs. They contain enzymes that physically slice apart invading pathogens or break down old, obsolete cellular components so they can be reused. Alongside them are peroxisomes, which serve a specialized chemical function. They neutralize and detoxify hazardous substances that build up naturally during normal cellular operations, like hydrogen peroxide. The elegant part of this system is where these defense mechanisms originate. The destructive enzymes loaded inside the lysosomes are actually just specialized proteins. They were built entirely by the very same nucleus to Golgi pipeline we traced a minute ago. Cellular sanitation is an active, ongoing defense grid forged and constantly restocked by the manufacturing pipeline it exists to protect. That brings up a final logistical question. When the Golgi ships a vesicle, or a lysosome hunts a pathogen, how do they actually travel from place to place without just floating aimlessly in the cellular fluid? The cell fluid is heavily organized by the cytoskeleton. Specifically, long fibers called microtubules originate from the center of the cell and lock every single organelle firmly in place. These fibers act as rigid scaffolding and dedicated internal highways for vesicles to travel along. Other structural proteins form intermediate filaments that push outward against the inside of the cell membrane, providing the cell its specific shape and keeping it from collapsing in on itself. This internal structure also extends to the outside world. In some cells, bundles of microtubules push through the membrane to form cilia that sweep mucus out of our lungs. Like the sperm cells shown in this footage, they form a single long flagellum, spinning like a motor to propel the cell forward through fluid. The cell's internal skeleton dictates the exact physical coordinates of every internal delivery and drives the mechanical movement of the cell itself. When we pull back to look at the entire cell operating at once, the old textbook diagram falls away. Instead of a bag of floating shapes, we see a buzzing, high-speed biological metropolis. DNA writes the instruction code, ribosomes assemble the physical structure, the Golgi apparatus boxes it up, the mitochondria powers the transit system, and the cytoskeleton provides the roads to move it. Life at the microscopic level does not happen because a cell simply possesses a checklist of correct organelles. It happens entirely through the uninterrupted, sequential flow of physical actions passed between them. Every time a human takes a breath, solves a math problem, or moves a muscle, they are relying entirely on trillions of these interconnected microscopic pipelines, executing their duties in perfect, unbroken synchronization.